Hello, I'm Maria Williams with the Collaborative on Health and the Environment. CHE enjoys bringing you the latest environmental health science through our partnership calls and webinars, science serves, publications, and social media. I would like to welcome everyone to today's CHE EDC Strategies Partnership webinar, which is titled Evaluation of PFAS for Potential Health Effects. Our moderator today is Jerry Heindel, Director of the Program on Endocrine Disruption Strategies at Commonweal. We will leave time following the presentation for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentation, our moderator will read out questions for our speaker to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speaker. This webinar is scheduled to last for 30 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Jerry.
Thank you, and good afternoon, everyone. I'm Jerry Heindel, and I'd like to welcome you to today's webinar. Today's topic, perfluorinated chemicals, seems to be the hottest topic in the endocrine disruptor area. And to discuss this hot topic, we have with us today Dr. Suzanne Fenton from the NIEHS and National Toxicology Program. Sue earned her MS and PhD from the University of Wisconsin-Madison. Go Badgers. Her postdoctoral studies were conducted at the UNC Chapel Hill Lineberger Cancer Center. And she did research at the US EPA's Reproductive Toxicology Division for 11 years. And then in 2009, she joined the National Institute of Environmental Health Sciences in their National Toxicology Program Research Labs. She's currently the group leader for Reproductive Endocrinology in the National Toxicology Program Laboratory. And she and her staff and trainees have published numerous manuscripts examining early life chemical exposures, including perfluorinated chemicals that lead to persistent developmental or functional changes over the life course. She has received several NIH and EPA-based science awards for her work on PFOA, including the fact that she was the first paper to show that PFOA was an obesogen. So we're pleased to have Sue with us today to update us on the science of perfluorinated chemicals and tell us a little bit about her current research in this area. So welcome, Sue.
Thank you, Jerry, and thanks for joining, everyone. Happy New Year. We're going to kick this off real quick because I know there will be questions. So I just want to make sure we're all on the same page when we're talking about per and polyfluoroalkyl substances, or PFAS. They used to be called PFCs. We kind of switched this PFAS name more recently for good reason. This is a group of compounds that now numbers maybe over 5,000, according to the dashboard at the EPA. These are non-stick, friction repellent, water, grease, and stain resistant type chemicals used in Teflon, Scotchgard, Gore-Tex, cosmetic industry, ski wax, food contact surfaces, and they're also used in aqueous film forming foam or firefighting foam to put out petroleum-based fires. So this is because it's used in so many ways and it's so persistent in the environment. Numerous military installations, airports, fire training sites are contaminated with PFAS now, and it's gotten into the water systems there. And so these chemicals are high interest to many government agencies and states for the safety of all their residents. PFOA and PFAS are probably the most well-known of these PFAS. We can be exposed to these in a variety of ways. today I'm going to kind of focus on ingestion or via drinking water or food contact surfaces, things like that, as a primary route, because that's what's been used a lot in the animal studies and that I'm going to be talking about. But there are numerous ways that we can be exposed, and a lot of those are shown here. So PFOA and PFAS are not produced in the US anymore, so who cares, right? Well, a lot of people should care because they're still detected in water systems throughout the United States, shown here on the left is PFAS and PFOA on the right, at levels that may not be safe for us to be drinking this stuff in our water. These are what we'd call legacy compounds. One, because they're not produced anymore, but we still have them around. But you know, who wants that kind of legacy is still up for question. But most of this contamination is associated with manufacturing sites. So PFOA and PFAS are definitely the two PFAS that we have the most information on in terms of how much people have been exposed over time and what their health effects might be. And as these have become eliminated in the United States in production, the levels in human serum have dropped. But as they've dropped, others have increased. And we may be seeing that increase over the next decade or so. So what's, unfortunately, the number of types of PFAS in our serum is probably increasing, as well as the level of each of some of those chemicals. So for instance, PFHXS might be more prevalent now than it was 10 years ago, whereas PFOA and PFAS are decreasing. But one of the problems with all of that is, you know, there's so many different types of PFAS now, and they're falling into some of these different classes. We know a lot about some. So the top arrow shows PFOA and some of the other legacy chemicals, where there's numbers of many articles that have been published on the potential health effects of these chemicals, and maybe their metabolism, their half-life, that kind of thing. But then there's others that are maybe in the PFESA class, which is in the middle, in that middle arrow, where we don't know very much about them. There's very little published. Now that might be changing really rapidly, but right now we know very little about their potential health effects. One of the kind of new chemicals that we don't know a lot about is, I'm going to call it GenX. It's shown here as a red bar in the top. Its structure shown on the left. This chemical is produced by Chemours and is been contaminating the Cape Fear River from the fluorochemical manufacturing plant in Fayetteville, North Carolina, which unfortunately polluted the water in the Wilmington, North Carolina area in the Cape Fear River Basin. Now, other PFAS are shown here. They're legacy PFAS. They're more in compartment A, which is at the top of the screen, where UNC, Chapel Hill, and NIEHS are near there. But so one of the points here is that we don't know a lot about GenX. It was measured at very high levels in the water in the Wilmington area, which has, since then, there's been remediation to decrease the amount. The state of North Carolina took action very quickly to limit the amount that could be in the water, and Chemours also took action and stopped dumping the chemical into the water. But you know, Community A's legacy compounds are things that have been produced over many years. They might be in the soil and in the aquifers and things like that, and they may not go away for a very long time. But those legacy chemicals are shown at the top here. Whereas if we take a closer look at GenX, GenX is only one of the many problems in the Cape Fear River Basin, because GenX is now shown in this figure on the far left as this tiny little red stripe. Well, you saw how big it was in comparison to the legacy chemicals on the previous slide. In this slide, you really get an idea of the fact that there are a lot of other PFAS in the water, in that area that we know absolutely nothing about. In fact, I think that it was difficult to figure out the names for some of these. The one in the blue with the blue box around it, I think might have been called the wrong thing, you know, when this paper first came out. I think now the authors might refer to it as something different. But the ones in the red box, we know, are still in the water in the Cape Fear area, thanks to the work of Jane Hoppin at NC State and some of her colleagues, and Mark Striner at the EPA, of course, and Detlef Knappe at NC State, who were involved in this initial discovery. But my point here is that these other chemicals, the PFO2, HXA, PFMOAA, we know very, very little about them. They're actually not even registered chemicals. These are degradates or byproducts of processing. And so these aren't actually things that had to have any health testing done on them, yet they're present in such high levels that they couldn't even be quantitated in this paper, even though this semi-quantitative numbers are shown in the x-axis. So I want to now transition a little bit to some of the work that we're doing within the NTP, and that is based on a lot of the work on PFAS and PFOA. We kind of have had to choose some target tissues, cells to work with, and a lot of that evidence in epidemiology and rodent studies on PFOA is what we base those selections on. So PFOA is actually associated with lower birth weight in infants in both humans and mice. So through a meta-analysis, really nice work done between the UCSF and EPA, they demonstrated that both in humans and mice, there is a lower birth weight caused by PFOA. And they can tell you exactly how much based on how much their increased exposure they might have, or at least a predictive amount. There's also an accelerated weight gain in prenatally exposed mice and humans. So when exposure was prenatal or during early life, both mice and human studies have shown evidence for accelerated weight gain as young adults. Yes, there are really young adult mice. So also, altered cholesterol levels are kind of a classic outcome for the PFAS class. Most PFAS are shown to alter cholesterol levels, and the same is true for the thyroid. Most chemicals in this PFAS class have been shown to alter thyroid function in some way. It may not be exactly the same outcomes in humans versus rodents, but the thyroid does seem to be a target. Kidney and testis cancer have been demonstrated in humans and the rats, and immune system suppression, particularly specific outcomes, have been shown to be associated with PFOA exposure through an OHAT systematic review that was published a year or so ago. And in the C8 Science panel, they were found probable cause for ulcerative colitis and immunization malfunction. So gestational hypertension is also kind of an outcome in humans that is there's probable cause for, and that's something I'm going to get back to in a little bit in our current work. But the mammary gland of human and mice have been shown to be affected either through delayed breast development during puberty and in the inability to nurse as they'd like to in mice and in humans. So in the NTP, our REACT program, which is Responsive Evaluation and Assessment of Chemical Toxicity, we've kind of used this approach for other environmental emergencies like spills and things like that. Well, we're taking that kind of a REACT approach with the PFAS group of chemicals. We want to try to provide enough targeted information that other agencies and institutes or states can make timely decisions. And we're really trying to focus on potential health outcomes and provide information that they can immediately use. So to do that, we're taking a multifaceted approach using in silico assessment, using lead scope QSAR, in vitro assessments based on some of these target tissues that I was just talking about, and then some more hypothesis-driven in vivo assessments. And one other thing that we've done is we've really enhanced communication with our research colleagues at other institutes, states, academia. We're trying to be very transparent in what we're doing and trying to stay in touch with as many people that are also doing similar work as possible. So we're not repeating efforts or wasting any funding on any of these endpoints. You can see here on the left-hand side the endpoints of interest that we've kind of chosen. The adipose tissue, liver, immunotox, placenta, mammary gland, kidney, and embryoid body models are currently either being developed or currently being used. And we're mostly doing a lot of this in 384 well plate models. So we're trying to do it as high throughput as possible so that numerous PFAS can be evaluated at once. And all of this is being done in a blinded fashion. So these are numbered by someone else, and we're evaluating them and then unblinding them after we've posted the data. One of the reasons we're looking at the liver in particular is because gestational exposure in mice, in CD1 mice, by 90 days of age, we can already start to see hepatocellular hypertrophy in a dose-responsive manner. So if you look at on the left panel in control diet, for instance, this comes from a paper that did a lot more than what I'm talking about today, but although we didn't see any of these effects by postnatal day 21, in 90-day-old animals, a dose-responsive effect of hepatocellular hypertrophy was evident in these animals. And in those that received the 1 milligram per kilogram dose, and remember, this is just gestational exposure, and most of this exposure was gone by the time we evaluated these animals. You can see the hepatocellular hypertrophy in these transmission electron micrographs on the bottom. In yellow, you can see the mitochondria. So we think that the mitochondria increased fission and fusion is part of what's driving the hepatocellular hypertrophy. And we see increased mitocellular DNA content, mitochondrial DNA content also in these animals. In the mammary gland, we also see persistent, lifelong effects of 1 mg per kg PFOA exposure when it's just gestational. So again, these are just gestational exposures. We've looked at 18 months with the high dose of PFOA, and at that high dose, we see stromal and epithelial hyperplasia. And in younger animals, but adult animals at postnatal day 84 on the bottom, we're definitely seeing disrupted development of the mammary gland that's persistent throughout the life of those animals. And although we have not published all of this work yet, we have pretty good evidence that this is not a PPAR alpha driven response. Same with the liver response previous to this. The mitochondrial response was prominent, and we did not see peroxisome proliferation in the liver. And in the mammary gland, we specifically evaluated for PPAR alpha. We saw changes in PPAR gamma that were pretty striking and significant, but right here what I'm showing is estrogen receptor alpha, and the fact that we're really, by exposing to PFOA during gestation, we really downregulate the ER alpha in the epithelium of the mammary gland, which is important in the ductal development of the gland to begin with. But another thing that we note here is this unusual fat pad. So in the treated animals, and we've noticed this in every study we've ever done, and we're doing some currently, and we're seeing it there too, the PFOA really affects the fat. The mammary fat pad here is disrupted. We see a lot smaller cells and a lot more cells, period. So cells other than the epithelium are responding to PFOA, but the epithelium is also responding in a way that's significant for the development of the gland. So it's interesting that the fat cells are affected here because we also noted several years ago that the animals exposed to PFOA during gestation got fat. So at the low doses. So at low doses of PFOA exposure, we're seeing fat mice like the one shown on the left here. In addition to that, we also see increased serum leptin and serum insulin, which we don't see at higher doses. Well, interesting enough, that's also kind of being seen in human populations that look at people who were exposed during gestation or early in life when the differentiation of the fat is occurring. So in our lab currently, Harley Cope has been working with 3T3L1 cells, which are pre-adipocytes, in culture in sort of a high resolution imaging. She can show that Gen X actually increases the number of fats, the adipocytes present in a dose-responsive manner, which is shown in the pictures on the right, and in the upper right figure with the blue label, we're seeing some significant increase in the number of total cells following Gen X exposure. These cells this increase in cells containing lipid increases the number of lipid droplets. So if we were to evaluate this in a standard way, we probably wouldn't see any effect here. But when you actually look at the number of cells and the number of lipid-containing cells, we're definitely seeing an increase that is dose-responsive. So one of the other issues that we're addressing is the issue of mixtures, because this really is a mixtures issue. So two other projects that we're working on in collaboration with other government agencies are looking at AFFF. We're actually testing some of the AFFF that are currently on the market that are not supposed to contain PFOA and PFAS. We're looking at what do they contain, what do they do in multiple cell types, what signals are they sending, and what fraction of the AFFF confers the activity. So is it the inert ingredients or is it the perfluorinated ingredients that are important? We're also trying to test water concentrates from the Cape Fear River Basin before and after the tap was shut off at Chemours for the effect of those chemicals as a mixture. And all of this work should be published this year. But I want to tell you about one last thing. So we have to be a little faster and think differently about how we're going to do our in vivo studies from here on out. So we have a lot of things that we're thinking about and talking about as models, but I'm going to talk to you a little bit about one hypothesis-driven mouse study that we have almost completed. Bevan Blake is doing this work. She's looking at the effects of PFOA and Gen X from embryonic day 1.5 to either 11.5 or 17.5 for effects on fetal weight gain, and really in particular looking at the placenta as the target for these effects. So we're really thinking about this gestational hypertension, and is there an effect at the level of the placenta, either in terms of how it's developed or what it looks like or the blood flow or any of those things. Those are kind of what we're looking for. So what we found in a nutshell in the moms is that there is an increase in weight gain over the pregnancy that is specifically related to the low Gen X, high Gen X, and PFOA doses. And that there's also, and we expected this, an increase in the relative liver weight. So this is the liver weight considering their body weight at both 11.5 and 17.5, at day 17.5. And there are 11 to 13 liters in each of these dose groups. She actually worked with Susan Elmore, Beth Mahler, and Brittany Scott here at the NIEHS to collect 3 fetuses per group. We determined the sex of each of those. The placentas were frozen. So here you can see in the right-hand side of each of these panels, the red line that goes from the snout to the tail. That's how we were measuring their length. Each of these individual animals was weighed, or fetuses was weighed, and we show some of that data here. So at E17.5, the high dose PFOA, and let me just say that this analysis was done taking the litter size into account. So these are a little bit more extravagant analyses, taking into account the litter size, and that is shown as the top variable on each panel. So you can see at the top that litter size does have an effect also on fetal weight. But anyhow, high PFOA has been shown before and is shown now to affect fetal weight in a negative way, in an adverse way. High Gen X and high PFOA actually increased the placental weight. And when we take both of those into consideration and look at the fetal placental weight ratio, which is something that's used in humans to assess adverse outcomes in a birth situation, we're finding that both the high dose of Gen X and the high dose of PFOA adversely affected these outcomes. We also have taken those placenta that were frozen and at collection and looked at them using NanoString with kind of a targeted group of genes. And we're finding that there's a little bit of a difference between the high and low PFOA groups in terms of gene signaling, whereas we're not seeing very much difference between high and low dose Gen X. But when you compare the top panel and the bottom panel, you'll see that there's only a couple genes in common. So there really are some differences in the gene pathways that are being triggered by these two different chemicals, yet they have some similar outcomes. So a lot of the transcriptomics are incomplete at this point, and we're going to be continuing those over this year. But in summary and in closing, I just want to say that this is really a challenging group of compounds that can be replaced without knowledge to the consumer at any time. So the more information we have on the more of these compounds will be useful. The half-lives and metabolism of most of these compounds are not known. I would say there's probably a dozen where we actually have some decent information. And I have a typo here. I want to try to say that there are differences by strain within a species. There are some differences. We've shown that in mice. And there are differences in the metabolism of these compounds by sex, in the rats specifically. We really need some modern tools for testing this class of chemicals. Transcriptomics, metabolomics, and high throughput screening are going to be useful, but we really need some good 3D models that have developmental stage incorporated in them. Thyroid and kidney models are needed for us to really understand the health effects of these chemicals. And honestly, the mode and mechanism of action of very few of these chemicals is understood. And it probably differs by tissue, like it's a tissue-specific, potentially, mode of action. And I don't think we should hang our hat on PPAR alpha, because the studies that have actually included human relevant exposures, most of which we don't know either, are not showing PPAR alpha to be very important. So with that, I just want to end. I think we have a lot of work to do, but I want to thank the people who I've been working with to do this work, and I'll take questions.
Okay, thank you very much. That was a terrific overview, both of the field and your own and NTP research. So we do have a few questions. So the first question really is, how's the NTP really trying to prioritize these 5,000 chemicals? I'm sure they're not all exposed to humans, but you only talked about a couple. How many are you actually working on?
Right. So currently we're screening a group of 45 and that we're almost finished with. So we should be publishing that within the next few months. We also are involved in studying another group of 75, which the EPA just published a paper in the EPA and other colleagues too, because Mike DeVito is included on that. Just published a paper in EHP like this week, kind of describing those 75 chemicals and how they prioritize them. So we do have access to that group of 75. But then also, we're trying to get our hands on the chemicals that we know people are actually being exposed to. So that's kind of how we're prioritizing at this point. Anything that we know that people are being exposed to, we're trying to get our hands on to get it in-house to test. So that's our first line of priority. We also will take into consideration the lead scope data that's being generated, the in silico work. We'll take that into consideration also to increase the number of things that we're testing.
Okay, thank you. So another question has to do with, you know, if you don't live by the Cape Fear River area, where is your exposure really coming from?
Lots of places, unfortunately. A lot of places across the country have been exposed, have had their water contaminated through the use of AFFF. That's not going away anytime soon because some of those are really high exposures. So for instance, if they're near an Air Force base or something like that, there may be some really high levels in water sources there. Otherwise, they're point source contamination from industry, and it's all across the United States. So on the third slide that I talked about, those two maps for the United States, those are available online, and you can look really closely at where they are. But I think there's going to be a new UCMR done in a year or so, or soon, I heard, that will help us understand all the different areas across the United States that are affected. Okay,
thanks. We're getting a lot of questions. We won't be able to answer them all. A lot of them are quite similar. I'm trying to go through. So are you looking at PPAR alpha, since that plays an important role in cholesterol?
We're using transcriptomics approach that are like whole genome wide approaches right now. So if in fact that pathway looks like it's significant for a certain tissue target, we would certainly know that. We're not exactly studying PPAR, but for the specific tissues that I talked about where we have some mechanistic data, we did definitely look specifically at PPAR alpha, and it didn't seem to be a significant player in those situations. But again, this is developmental exposure to lower level PFOA, so
okay.
You know.
So one question is, technically, how do you deal with those chemicals in the lab, you know, for lab contamination, since they're kind of everywhere? What do you have to do about that?
Yeah, so we mix our chemicals in a very specific place, and like in a hood space, and they don't come out of there unless they're in methanol. So we've really gone away from the use of DMSO for these things in culture because Gen X in DMSO gets degraded, apparently. So it may look like there's no effect, but it's because it's been sitting in DMSO for a while. Apparently that's been pretty widely known by chemours, but we only recently discovered it because we had no effect one day and we had effects of it previously. So now we bring up all of our chemicals in methanol, and we try to do everything in hood space that's cleaned very carefully after every use.
Okay, and another question has to do with the doses. So how does 1 milligram per kilogram match human exposures, and are those considered really low dose effects?
Yeah. So some work that we did in the mouse mammary gland in particular, we actually measured the internal dose in the animals, and it compares pretty well to highly exposed kids in the C8 Science Panel study. So at 21 days, when those animals are weaned, there is still PFOA detectable in the offspring that were exposed to 1 mg per kg. But by the time they're about 6 weeks old, we can't detect that anymore. So at the time when we were actually seeing the effects, the serum levels in those animals are definitely similar to serum levels that have been reported in highly exposed communities, but not your average person.
Okay, so have you looked at lower levels to try to see if there's a threshold or anything?
Yeah, yeah, actually, at 0.01 mg per kg, we could still see effects in the mammary gland, and we could still see effects on obesity. So again, both of those have fat cells involved in them. So maybe fat cells are really sensitive to this chemical. We also, if you were looking carefully on the first part of that liver slide that I showed, we did see some hepatocellular hypertrophy at 90 days also. But like at day 21, we couldn't even detect PFOA in the serum of those animals at that level. We can't detect it above control. So we know that's kind of like our low L. We don't know what the no L is for PFOA, but you know, in some of the studies that we have that we're going to be doing, we're definitely going to be trying to get a no L for some of these effects.
Okay, last question, because we're kind of over time here. Are you guys doing any testing on biotransformation or toxicity of metabolites and stuff?
Yeah, that's a really good question. There's some challenges in working with these chemicals. So one of the challenges in doing that work is that we're not exactly sure. Well, let me just say, we're doing some studies right now on metabolism of these chemicals. Are they metabolized by metabolically competent, like say, liver cells? So we are doing those types of studies. So one, are they metabolized? We want to know that, because for some of these chemicals, we don't know. Two, when we put them in cell culture, we're not sure that all of it actually gets to the cells, or if it forms a layer. I mean, because these are kind of like, you have to think about this class of compounds. It's, you know, they might be forming a film. We don't know exactly if they're getting into the cells. So we're actually working on some of that also. I know there are also people at the EPA that are specifically working on some of this. So stay tuned, I guess, on that note, because that's really a complicated piece of all of this and what makes this class of chemicals difficult to work with.
Okay, Maria, we have 25 questions, so we can't answer all the questions. So how does that work? Will Sue be able to answer these questions and post them somewhere, or what do you want to do?
We can email the questions to Sue and she can respond to them individually. Does that sound good, Sue?
Sure.
Okay, great. All right. Thank you. Okay, and thank you very much.
Thank you, everyone.
Jerry. We are approaching the end of today's webinar. A video recording will be available on the CHE website soon, and tomorrow you will receive an email containing a link to the recording. CHE's next webinar from the CHE Boston University Superfund Research Program Partnership will take place next Thursday, January 24th, and is titled Climate Change and Toxic Hazards Before the Storm. To learn more into RSVP, please visit our website at healthandenvironment.org. CHE's next webinar from the CHE EDC Strategies Partnership will take place on Wednesday, February 20th, and will feature an interview with Dr. Leonardo Tersande about his new book, Sicker, Fatter, Poorer, The Urgent Threat of Hormone-Disrupting Chemicals on Our Health and Future and What We Can Do About It. Details will be available on our website soon. If you are new to CHE and would like to stay updated about upcoming events and more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website. Additionally, if you appreciate these CHE partnership webinars, bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank Dr. Fenton for taking the time to talk with us today and Jerry for his excellent moderation. Thank you so much for joining us and have a great day.